Right, so these are your video directions for this genetics lab, okay? So the first thing, it says in this lab you're going to test out genes A, B, and C and see if you can figure out what each gene does to the bacteria, okay? This is the link to the lab right here in blue. And you're going to be filling in this table first off. That'll be the easiest thing for you to do is start filling out this table. So on the lab, you'll have the option to turn on different genes. And I've given you a place to put those observations so that they can be organized and you can go back and look at them. Once you have filled in that table, you're going to scroll down and answer questions 1 through 7. Make sure you answered all of these questions, okay? So really quickly, I'm going to show you what it looks like when you click on this lab. All right, so this is what it's going to pop up with to start off with. You're going to click this big red next arrow down here. It's going to tell you step 1, 2, and 3. It's going to show you how to use some of this stuff, but I'm going to show you the same thing on this video. All right, so these are my options here. I can turn gene A, B, and C off and on. I click on it and it changes it. So it's like a light switch. It can be either off or on. That's how you change it. Right, once I've turned on the gene that I'm looking at, because on that table it says only gene A is on, that's that first section on the table, I am going to click this button that says make a mutant. And then I'm going to have 3 ways to look at this bacteria, okay? The culture is how we grow the bacteria, and it grows on what we call media. So rich media has got a bunch of food and all kinds of other stuff on here for that bacteria to live off of. And then the limited media is only going to have food on there. It's not going to have a whole bunch of other stuff for that bacteria to eat. Okay. So I'm going to come over here to where it says light microscope. This is going to let me look at that bacteria a lot closer and see what it's doing. So I'm going to click on light microscope and I can see the bacteria are on here. That's what these little things are on your screen. Those are the bacteria. And this green right here is food. They don't tell you what the food is. This is just food. So you can think of it however you want to, okay? So I'm going to put in my table that the bacteria are wiggling, but they're not really moving around. They're not going from one side of the dish to the other, okay? So that is my observation for just gene A. So I will come on here. I can type in that information. All right. So the bacteria are wiggling, but not moving around. Oops. Not moving around the dish. All right. And then I'm going to go back. It says the next one, it says only gene B is on. So I'm going to go back to my lab. I'm going to tell it I want to make a different strain. And then I'm going to make sure that gene B is the only one that's on. Gene A is off and gene C is off. Gene B is the only one that's on. And then I'm going to tell it I want to make a mutant. And I'm going to do the same thing. I'm going to look at the culture. I will come to the light microscope. And then, oh, forgot to show you this one last time. Once you're in this, you can see what it's doing under the light microscope. You can come down here under the electron microscope and see more details about what the bacteria is shaped like, okay? So this is something else you can write about on your table. So I'm going to go back and do that for gene A again. So just gene A is on. I'm going to come look at it under the electron microscope, and I'm going to make observations about the bacteria here. So the bacteria wiggling, we're not over in the dish. All right. So what the bacteria looks like when we look in a lot closer, it says the bacteria is gray and oblong. That just means it's kind of like a jelly bean shape. Okay. So that's what I observe about the way the electron microscope sees. It sees it a lot closer. It gives us a lot more detail than the regular light microscope. Okay. And that is something I do want you to add into that table. So you need to tell me what they're doing under the light microscope. And then I want you to look at the electron microscope and tell me what they're shaped like, because that's going to be something that's going to help you understand what these genes do for that bacteria. Okay. And once I've filled in this table, I want you to come down here and answer questions 1 through 7. If you need help on this, or if you have questions or you get stuck or confused, please ask. That way I can come and help you.